Hello everyone, hope that you're good. Today I'll be talking about hepatitis B, or inflammation of the liver, which is quite common in Pakistan and other parts of the world. Um, I would talk about certain aspects like why different antivirals are not working, although people are using these antivirals for years and years with no positive outcome. And I will also talk about its replication strategy, that how this virus is replicating inside the liver cells, um, how it is different from hepatitis C virus and other viruses. And I also would talk about some new promising technologies like the therapeutic vaccine, which has been developed, which is used both as a therapy and as a vaccine, and has given promising results. And it's a good news for people who are already infected with hepatitis B, that now they can be treated with this therapeutic vaccine. Um, a vaccine is usually used for prophylaxis or prevention of disease, but this particular vaccine would be used in people who are already infected. And this is quite promising and a good news for such people. If you look at the burden of the disease, it's quite significant, more than hepatitis C virus, more than other viruses like HIV, etc. About 260 million people are infected with this particular virus chronically around the world, and about 880,000 people die because of complications due to infection with hepatitis B virus, which include this liver failure or hepatocellular carcinoma, or HCC, which is called the liver cancer. So it's quite a significant burden. Currently, no curative therapy is available. The therapies available to date inhibit the virus replication but need to be given for longer periods of time. And these longer these longer duration therapies are not well tolerated by many people, and they do not result in the eradication of virus from the system because of the inherent, because of certain traits of this virus, for instance, getting into the nucleus and staying there for a longer period of time. This is something peculiar to this virus and some other viruses. As long as infected people cannot form an adequate immune response, the virus will survive. Immunity has a great deal to do with this particular conditions and with other condition as well, because immunity, if your own immunity is not working, your immunity is suppressed, which happens in the case of disinfection due to overproduction of these viral proteins, then therapies would not work properly. So the antiviral therapies like interferon-based therapies and other therapies are not working quite effectively. So the antiviral therapies, which are effective against other viruses, are not effective here in the case of hepatitis B virus. I will talk about it in a bit detail in other slides. Vaccine is not effective in about 5% of the people, although this vaccine is available since 1981. But this vaccine probably was the first recombinant vaccines against any virus. And over the past 4 decades, we have been using this vaccine because we couldn't make herd immunity against this particular virus, the hepatitis B virus. And still, it is quite prevalent and is causing significant burden of infections around the world. It's because of several reasons, including this poor immune response in people, and including certain other aspects like the escape mutants or mutations which are taking place in the viral strains of hepatitis B virus, and that due to that, the vaccine is not quite effective in such people. We will also talk about it later. Who are the prime target? Children are the prime target. They most often would develop chronic hepatitis B infection. It happens in the case of these uh, if somebody is infected during the childhood, or if somebody has got this infection from their mother to child transmission is quite common. So the children most probably develop chronic infection. Chronic infection of hepatitis B is dangerous because it leads to cirrhosis and hepatocellular carcinoma, liver cancer in these people. But with growing age, the chances of developing chronic infection is just minimized. Um, for the common audience, what is hepatitis B? It's inflammation of the liver due to hepatitis B virus. Inflammation of the liver could be caused not only by viruses but also by certain bacteria, for example, certain chemicals like alcohol, etc., some other toxic substances. But we are particularly talking about hepatitis B here. So this particular virus, when it infects the liver cell, it can cause this inflammatory situation there. What are its associated complications? This infection can lead to inflammatory situation, and it would later cause, fibrosis, cirrhosis, and the liver cancer or hepatocellular carcinoma. So this is what happens in the case of this particular infection. This is the course of infection. This inflammatory situation, which is generated here in the liver cells due to infection with the virus, may be acute, which would last only for 6 months, and then it would be resolved. In majority of the people, it happens like this, and it can become chronic. Just like I told you, in children it can become chronic. There are maximum chances, and it will last for years and years. The hepatitis B virus would just uh, enter the liver cells and then enter the nucleus of the liver of particular liver cells, and it will reside there in the new liver cells for you know it would reside there and would cause this inflammatory situation for years and years. And that leads to scar tissue formation, which is known as fibrosis. And the scar tissue formation or fibrosis at advanced stages would lead to this cirrhosis, where the entire liver tissue would be damaged. It would become fibrotic, and the channels of the liver would be blocked. And this is the time that excessive pressure, because everything that is coming entering into your body has to pass through liver, so there's a lot of pressure on the liver, and this liquid would just ooze out of the liver into the abdominal cavity when there is no access, when the accesses or the channels of the liver are blocked due to cirrhosis. So this is a normal liver that you see here, and this is the cirrhotic liver. The liver tissue is amazing. It's the biggest internal organ, and it is involved in 500 vital functions of your body, including clearing these toxic substances from the blood, including production of different immunoglobulins or antibodies. So lots of different things are going here inside the liver. And this job is performed by the liver cells. But when the liver cells are damaged, just like this, they can't do any job. It means that anybody's productions, clearance of toxic substances from your body and lots of different functions would not be possible to occur here in the liver. So it means it would generate a situation where there would be a lot of toxicity uh, uh, would be developed inside your body, and that would lead to severe consequences. You would start feeling these symptoms from severe fatigue to fluid retention and mental confusion when your liver becomes cirrhotic. If one third of your liver is okay, you probably would not feel any symptom. It takes a lot of time. It may take, 10 to 20 years for the livers to become completely cirrhotic. But it happens slowly and gradually when somebody is chronically infected. In some cases, the infection would just not be detectable when you are doing the blood test or antibody-based test or nucleic acid-based test, because of the virus, not which is not present in ample quantity in the blood by that time. It can cause occult infection as well. It would remain there inside, the hepatocytes or inside the liver cells, And as your immune system is, going down, the virus again would start replicating and causing the problem. That happens in many cases. Um, this virus is important in some other aspects, like the history of modern research in viral hepatitis began in the year 1963 when Nobel Prize winner B.S. Bloomberg reported for the first time publicly on the discovery of a new antigen named Australia antigen. It was quite a desperation, on the part of the scientific community because they didn't know about the cause of the disease, although this disease was described centuries ago, but nobody knew what actually was causing this particular disease. And this Bloomberg was the first scientist who described that there are certain antigens in the plasma of patients who are suffering from hepatitis B, which are not present in other patients. And he just, termed it Australia antigen. And later on, we came to know that this Australia antigen actually was the hepatitis B virus surface antigens. We now know more about hepatitis B surface antigen, which is quite important. It's involved in a lot of immune suppression in the host because lots of HBsAg is produced there and it just comes out of the hepatocytes. It's not only making the complete very young, but some free HBsAgs are also coming out into the open, into the blood or plasma, and it is doing lots of immune suppression. Is involved in the development of these severe phenotypes of the disease, including hepatocellular carcinoma or cirrhosis. Another very special aspect of hepatitis B virus research is the human experimentation because uh, it didn't happen in the case of other viruses because ethical protocol were established by those times. But by the time people were desperate to know about the cause of hepatitis B, they even experimented with prisoners of wars and mentally ill patients. Uh, and uh, Although it's quite unethical when you look at it from the ethical point of view, uh, by today's standards, so it's quite unethical. But in those times, in Second World War, it was that time period. People desperately wanted to know about the cause, and that's why they used these mentally ill patients and prisoners of wars for their research. And it was later in the 1970s when this infectious HPV variant, which is known as the Dane particle, was discovered by this scientist, David Dane, who was a British pathologist, and he was observing the plasma of patients of hepatitis B virus under the electron microscope, and he observed this complete Dane particle. And later on, it was discovered that hepatitis B surface antigen is actually part of the Dane particle. Now, this is $1 million question: of why antiviral therapies are not effective against HBV. It's because of certain reasons. For instance, one reason is that if you compare it with hepatitis C virus or other viruses, the hepatitis C virus is when it enters into the cell, it is entering into the cytoplasm, it's not going into the nucleus of the cell. On the other hand, when HBV or hepatitis B virus enters the hepatocyte of the liver cell, it doesn't stay in the cytoplasm. It has to get into the nucleus, so the core particle would enter into the nucleus, and in the nucleus, that its DNA is converted into covalently closed circular DNA or cccDNA. So the cccDNA is not accessible. It is quite a stable structure, not accessible to the antiviral machinery of your cell. And that's why it stays there for longer periods of time. And it happens in the case, mostly in the cases of HBV infection. It's the survival mechanism of HBV as well, that it gets into the nucleus and it's converted into cccDNA, which is quite stable, not accessible. So getting into the nucleus ensures its escape from the antiviral wing of the immune system. And on the other hand, overexpression of HB viral proteins suppress the immune system. As I told you about HBsAg, there are other viral proteins which are overexpressed. When the cccDNA is converted into these reading frames, these mRNAs of the viruses, Lots of these, proteins, just like the surface antigens, they are produced there, and some of them can be free uh, to come out of the hepatocytes and interact with different proteins inside the cell or outside these cells, and you can find abundance of these proteins in the plasma as well. So it will become clear from this sketch. This blue one, these are the hepatitis C virus, and these purple are the hepatitis B virus. When hepatitis B virus enters into the cell, it's a cell receptor mediated entry. It will attach to its receptor into the cell. The its entire its outer surface coat will be just removed, and the core particle would enter into the nucleus. Because the core particle is small, I would show you that in another slide. And here it's genome. The DNA would be converted into cccDNA, which it can be transcribed and translated later in the cytoplasm to make these particles, infectious particle or the tame particles to be released. While the hepatitis C virus, it's injecting its genome or the mRNA, which is its genome, into the cytoplasm. It would be directly be translated by the machinery, because the translational machinery is here in the cytoplasm. And these products are made here, and the genome also is replicated here in the endoplasmic reticulum, the web that is formed there in the ectoplasmic reticulum. And again, the replicated genomes in these protein products are assembled in the form of these virions, which are released. So you see the hepatitis C, it's not going into the nucleus. We're just completing all its replication, transcription, and translational process here in the cytoplasm and going out of the cell. But the hepatitis B is not like that. It is different. It's going into the nucleus. It's staying there for longer period of time. It's not accessible to certain elements of the antiviral system of your immune system to target. For instance, interferon. They are expressed here. The mRNA is formed here, and this mRNA is translated here to make these interferons or interferon stimulatory genes, making different products to block viral replication in the case of hepatitis C, but it cannot do it in the case of hepatitis B. Okay, so that is the difference between these two, that certain elements of the antiviral systems are not targeting this virus properly. And lots of different antiviral drugs which are effective against hepatitis C are not effective against hepatitis B. If you look at the particle, the hepatitis B virus infectious particle, you would see that at the surface you can see the surface antigen, which is known as HBsAg, that was we talked about. Then there is another circle here, which is the capsid. This gray color capsid that you see, this is composed of the core antigens. We call it HBcAg, or the core antigen. And there is envelope antigen. There's another layer here, which is called the envelope antigens. We call it the HBeAg. And there is a DNA polymerase which is attached here to the DNA. And apart from it, there also is a reverse transcriptase. So there are two enzymes, DNA polymerase and reverse transcriptase. These are two different enzymes doing different jobs, and they are already present inside the hepatitis B virus then particle or the infectious particle. Okay, this is the partially double-stranded DNA, to which on one side you can see this DNA polymerase is attached. So it's a partially double-stranded single copy circular DNA of about 3.2 kb. So 3.2 kb DNA is not a big size, but if you look at the tricks that this virus is playing on humans, in its survival rate inside the humans, you would come to know that how this virus would have evolved and how smart this virus is in order to, you know, counter or immune responses or dampen our immune responses with the help of these different proteins that we see here that is making the structure of this particular virus, or other proteins. Uh, these are actually not letting the immune system to be very active against it. Now, I told you about its entry into the nucleus. Why hepatitis C virus is not entering into the nucleus. This virus is getting into the nucleus. The capsid, actually, this capsid protein, which is making the core. This is the core particle which is inside the capsid. It include this DNA and these enzymes. This thing actually is entering into the nucleus because of its smaller size. It's about only 36 nanometer in size. And it is composed of these core proteins, which is the size of the core protein is 21 kD, and 240 core proteins make this entire structure of the capsid. So in some viruses of other genotypes, you can find more particles, more than 240 copies of the core protein, but in majority of them it's only 240 copies. If you look at the size of the entire particle is 42 to 46 nanometer, which cannot get to the nuclear pore complex, but 36 nanometer is a smaller size for this, core particle to enter through the nuclear pore complex. Something that gets into the nucleus should have the nuclear localization signals. If these are different peptide something, so the signals would just take them to the nucleus, and its size also matters, that its size should not be very large. If you look at how the genome is transcribed and different proteins are made, there are four overlapping open reading frames which are known as open reading frame. S, C, P, and X, for instance, S is for structural proteins. These open reading frames are making different structures and non-structural proteins of this particular virus. And all these proteins, are not only making these infectious particle of the virus, as I told you, they are playing other tricks as well, and they are also, uh, uh, you know, suppressing the immune system. How does HBV infect the liver cells? First, they would attach to the heparin sulfate proteoglycans, and then they are shifted to these specific receptors for hepatitis B, which are called NTCP. These specific receptors would dimerize, and they would take this particular virus inside. Endocytosis would take place here, as you can see. So the surface would just collapse with the plasma membrane of the cell, and the core particle would just go into the nucleus. So NTCP is a specific receptor for it. It's called sodium taurocholate co-transporting polypeptide, or NTCP, which is present on the surface of hepatocytes for the entry of this cell. Now this slide is very important. It describes the HBV life cycle or replication, and it starts with attachment of the virus with the liver cell. And for attachment, I told you about these proteoglycans and the NTCP. These receptors would dimerize and take the virus inside, and then, as you see here, the outer surface membrane is gone, just letting the core particles to enter into the nucleus. So the core particle is going to enter here. So there would be attachment is followed by internalization, this nuclear import. It's getting into the nucleus now. What happens here in the nucleus, that viral DNA polymerase make fully double-stranded DNA. As the genome of the hepatitis B virus particle is partially double-stranded. So once it gets into the nucleus, the viral DNA polymerase, which is present inside the nucleus here, would start making, copy of the DNA in order to make a full circle, or to, complete the partially double-stranded DNA into full or double-stranded DNA. And that full double-stranded DNA again would be converted into cccDNA. This covalently closed circular DNA is a very stable structure. If you look at it, it's a helical structure, looks like figure 8. and this can stay here in the nucleus for longer periods of time, and that is the problem. It's not accessible to many things, and it can stay there for longer times, playing with your immune system. Later on, this cccDNA would be converted into these four reading frames that I'm showing you here. I told you about the reading frames. It would be transcribed. In this transcription, for this transcription, the virus is not using its enzyme. The virus would use the host RNA polymerase. The transcription into pre-genomic mRNAs by host RNA polymerase would take place. So these are known as pre-genomic RNAs, which are transcribed from the cccDNA by the host RNA polymerase enzymes. And these pre-genomic mRNAs, these are actually from the four reading frames that I told you, and these are coding for all the structural and non-structural proteins. For instance, these are the capsid proteins here in orange color, and the red color are the DNA polymerase. The DNA polymerase core proteins and the surface antigens, all the proteins are encoded by these mRNAs. So once the DNA polymerase and the core proteins are made, the DNA polymerase is attached to one end of an mRNA, the pre-genomic mRNA, which has um, a packaging signal. So the DNA polymerase attaches to the packaging signal of pre-genomic RNA, and it's packaged in core particle. Once the DNA polymerase attaches to this structure, what happens? Uh, you know, when it attaches to the pre-genomic RNA, then these core particles start encircling these pre-genomic RNA and DNA polymerase assembly, and just making an immature core particle. And this gradually would mature as time passes, as reverse transcription now would start place, because reverse transcriptase is also there inside the core particle, and this reverse transcriptase would make genomic DNA, the plus and minus strain genomic DNA. So this partial DNA again can be recycled into the nucleus and could stay there, because certain copies should be there inside the nucleus, and others could be converted into fully functional HB virion or the DAN particles when it matures. Okay, because the surface proteins and other proteins that are made, they would also make different parts of this particle. They would just surround around the core particle, which is there, having partially double stranded DNA polymerase, reverse transcriptase, the envelope proteins, and the surface proteins, and all these things would come together to make this infectious particle. And as these proteins are made in excess, so some of these proteins can make these empty baryons. Some of them, the surface antigens, can, as such, get out of the cell into the plasma, and these proteins are a problem for immunity. So envelopment release is the last step which takes place, but these, you can say, proteins, they are, it's a strategy of the virus to suppress our immune system with the help of these proteins that it is making. And this is the therapeutic vaccine. So the basic theme of this therapeutic vaccine was the basic idea behind this effort was that large amount of hepatitis B virus proteins expressed in the liver prevent the body's immune system to defeat the virus, consequently preventing an effective therapy. Because of the excessive amount of these different proteins which are made here, viral proteins made in the liver, they are interacting with different partners of the immune system and other proteins, suppressing your immune system or T cell responses or natural killer cell responses. Now, that's why the body cannot completely thoroughly clear itself by just killing all the cells which are infected, or immune system is not working properly. So in this paper, and it just appeared in Gastroenterology in 2020, T. Mishler and his colleague, they devised a new strategy, a new vaccine, which is known as a therapeutic vaccine, which is used as a therapy. It means that you can use it after infection. If after somebody is infected with hepatitis B virus, this vaccine could be used as a both as a drug and as a vaccine. Normally, we use vaccines for prevention of disease, but this is used for cure. So what did they do? They used these small interfering RNAs. You know about the small interfering RNAs. These are short chain RNAs, and they target different mRNAs. So they designed these siRNAs to target these viral mRNAs. These reading frames are viral mRNAs, which are responsible for making viral proteins. So they made siRNAs for them, targeted them, and just destroyed them, and looked at the effect, that what would be the effect on this. If you can bring down the expression of the viral proteins, then you can deal with hepatitis B. You can control hepatitis B. So in the first thing that they did was to use siRNAs to target viral mRNAs so that their expression is lower. But at the same time, they observed that it was not sufficient because as these viral proteins target different components of our immune system, including the T cells and the natural killer cells. So they observed that the T cell inhibition was not reserved. The CD8 reverse, CD8 positive T cells, cytotoxic T cells, which are important for killing infected cells, virally infected cells, are suppressed by the viral proteins. But when they knocked down the viral proteins, they saw that this during the course of the disease, already the T cell population was significantly reduced. Inhibition of the CD8 positive T cells had taken place, which was not reversed even when they suppressed those proteins, viral proteins. So then they administered a vaccine. They made a vaccine. They called it TherVacB. Ther is for therapeutic, vac for vaccine, and B is for hepatitis B. They didn't describe that what is there in this vaccine, probably a recombinant vaccine with some other components or some motives to trigger your immune responses, your T cells and natural killer cell responses, along with making triggering these B cells to make very specific neutralizing antibodies efficiently. So they just, made this particular vaccines, and first they targeted the mRNAs with siRNAs, and later on they administered this particular vaccines, and they observed that whether hepatitis B is going to the hepatitis B virus is going to be contained or destroyed or not. So they were able to completely wipe out hepatitis B virus from the mouse model. Humanized mouse models were used, mouse models with human genes, which can reproduce the same kind of condition as you can observe in the case of human beings. So they completely eradicated the virus from these different mouse models that they use by this strategy of, you know, giving siRNA first to target the mRNAs of the virus, and then followed by this particular vaccine. So the therapeutic vaccine developed is indeed very promising, as it induces neutralizing antibodies and T cell responses. So it's not only affecting the B cells here, that it means that it would have some recombinant components because the neutralizing antibodies would be produced by the B cells, and the B cells, the memory B cells in your body would be clonally expanded in that case. So it would have some components of probably, some surface antigens and some other motives in order to trigger these cell-mediated and humoral responses. Both the components of the acquired immunity, the humoral immunity and the cell-mediated immunity, are increased here in this case. So although the invention has not been described entirely, but it would have components like that. Human trials of this vaccine are beginning in 2021, and would be quite promising because we did not have any therapy against hepatitis B would to completely eradicate the virus from the bodies of infected person. And this is for the first time that we can hope now that the virus would completely be eradicated from the bodies of the, and there is another discovery. This is probably the second last slide. Discovery of the memory natural killer cells. It's a very important discovery. You know, we knew about the memory B cells and memory T cells. The T cells and B cells, they can keep memory of different pathogens. But we didn't know that the natural killer cells, which are important cells of the innate immune system, or can also keep memory. This is quite significant here, because the memory cells that keep memory of pathogens, they recognize pathogens with different patterns, and when that pathogen infects that person again, these memory cells are clonally expanded and they mount a very robust response against that pathogen and neutralize the threat over there. But we didn't know that the natural killer cells can also do this particular job. So that's why the natural killer cells, this is very important that the natural killer cells have memory. We can use this information to improve the vaccines that we made against different viruses, and to make therapeutics or vaccines in such a way that this particular component, the memory of the natural killer cells, would be included in the formulations, and that would enhance our capability to deal with different pathogens quite effectively. And vaccines which were not working properly before would become more effective as a result of that, because of this particular condition, this information. This is a natural killer cell. This is the last slide. You can see that cytotoxicity is killing of the cell. It is killing tumor cells or virally infected cells by different mechanisms, including granule-mediated cytotoxicity, cytokine-mediated cytotoxicity, antibody-mediated cytotoxicity. So there are different, wings of this particular response of the natural killer cells to kill different infected cells by recognizing different infected cells, with the help of antibodies specific for certain antigens, or via granule mediated cytotoxicity or cytokine mediated cytotoxicity. So all these things are coming together. These natural killer cells are going there to kill the infected cells or the tumor cells. And the natural killer cells, as we now know, that they have memory as well. So in the case of memory, such tumor cells or virally infected cells would be very readily targeted. Lots of cells would be coming here. There would be expanded clonidine. They would just target the virally infected cells and deal with the threat effectively. So that is quite significant research, but due to this pandemic situation, the coronavirus situation, even some valuable research, didn't get the desired attention. I hope that you like the lecture. If you have any question, you can just um, comment on my lecture or the videos on GTV Education, and I would be very happy to respond. Thank you very much for your patience.